My name is Stefanie Severin, and I'm a senior Animal Biosciences student at the University of Delaware. This summer, I had the pleasure to work with Dr. Mark Parcells. My project focused on Marek's disease and how we can create a new and more efficient vaccine for chickens. Marek's disease, or MD, is actually the most common cancer in the animal kingdom, including humans. This cancer is caused by a highly contagious alpha herpes virus known as Marek's disease virus, or MDV. In chickens and turkeys, MD is characterized by paralysis, immune suppression, tumor development, and death. Birds spread this disease through the inhalation of dander or litter contaminated with feather follicles from infected birds. MDV is composed of 3 viruses: Mardivirus 1, Mardivirus 2, and herpes virus of turkeys. MDV1 strains are responsible for causing the cancer in infected chickens. There are existing vaccines that prevent mortality in the development of tumors. One of the most common vaccines is derived from HVT. However, it still permits birds to become infected and shed MDV1 field strains. Because of this, it is possible for a more virulent strain to evolve. Back in 2001, Doctors Parcells and Kung discovered that MDV encoded what looked like a viral version of the chicken interleukin-8 gene, and they called this VIL-8. So, what is the role of VIL-8 in all this? VIL-8 was previously believed to be homologous, or having similar function, to interleukin-8, which is a chemokine that attracts neutrophils when cells are infected by microbes. This was the first CXC identified in chickens. However, it was later revealed that VIL-8 was most likely an ortholog to a chemokine in chickens called CXCL13, which recruits B cells rather than neutrophils. In 2017, Herdel and researchers found that deleting VIL-8 from the MDV1 genome significantly reduced the ability of the virus to infect B cells early in infection. Therefore, we predict that adding this gene to the HVT vaccine strain will increase its replication by attracting more lymphocytes for infection. We will be attempting to create a recombinant vaccine, which means that it will contain a modified version of the HVT genome. We can use genetic engineering techniques to create a subunit vaccine including the gene encoding VIL-8. The first step will be to insert the sequence encoding VIL-8 into the virus. This virus will be placed into chicken cells via a process called transfection. Then, we will screen for the presence of VIL-8 using antibodies or various PCR reactions. We can examine the DNA at the molecular level to get an understanding of the genes that are present. Once we have determined that the product of VIL-8 is being produced, this DNA can be extracted. Through technology like nanodrop and gel electrophoresis, we can measure the concentration of DNA, obtain an estimate of the size of the product, and determine the purity of the sample. Nanodrop is used as the means of quantifying the concentration of the DNA after isolating it from a cell. These DNA samples can then be loaded onto a gel and go through a process called electrophoresis, which produces visible bands that travel different distances based on the size of the DNA fragments. This helps give us an idea of what the sequences in our DNA samples look like. When screening for VIL-8 at the microscopic level, here is what those cells may look like. We can use various wavelengths of light to look at different aspects of the cells. For example, the red fluorescent cells indicate they are positive for VIL-8, and the white fluorescent circles show the intranuclear proteins within the cell. MDV is one of the most prevalent infectious agents affecting poultry. Evolving strains with increased virulence require more effective vaccines. MDV is highly virulent, and the fact that the virus is still able to replicate and evolve within even vaccinated birds poses a threat of newer evolved strains with increased morbidity. As MDV can have devastating financial impacts on commercial flocks, a vaccine that induces a more robust immune response would be advantageous for poultry production. This summer research program has enabled me to explore my research interests and add new lab techniques to my toolkit. I have also built my critical thinking abilities from day-to-day problem solving. I am glad to have participated in this program.